Good morning, everybody. Happy Monday, April 27th. This is going to be our lecture video beginning our discussions on DNA. Right? We all started learning about DNA actually a long time ago when we were in biochemistry, and we learned the monomers and polymers of DNA. All right, so let's hop into it. All right, so DNA is a acronym for deoxyribonucleic acid. Right, very long name, hence why we just call it DNA. Right, and we learned that the monomer of DNA or nucleic acids were nucleotides. Right, and nucleotides are made up of three different things. All right, they have sugar, which is in the form of deoxyribose. All right, the OSE ending means it's a sugar. It has a phosphate group. All right, so it's actually, we're going to learn that it has a sugar phosphate backbone. And then it has nitrogenous bases, which are, it actually carries the genetic code or the genes, okay? All right. And so the nucleotides are going to be what we are pretty much focusing on when we start getting into the steps of DNA replication. All right. But before we get into actual replication, we need to talk about the history and discoveries of DNA and talk about some important scientists. All right. But just to go through this last parts, all right. The four names are here in different colors. There's thymine, adenine, guanine, and cytosine. Right? These are always going to have a rule. We're going to learn that this is actually Chargaff's rule. Okay, and notice how over on the right, whenever you see a C, you're going to see that it's paired with a G. All right, and whenever you see an A, it is paired with a T. Right? A's and T's go, and C's and T's go. All right. And what they're actually connected with is a hydrogen bond. And that's what this big H is here. All right. And so if you can wrap your head around the matching of just adenines pair with thymines and cytosines pair with guanines, DNA replication should be a little bit easy. And the biggest hiccup might be just from remembering some of the bigger names. All right. So let's go into some of the names. All right, so DNA is shaped like a double helix, so it's like a twisted ladder. All right, we all know what a straight up and down ladder looks like. Imagine if that was just to twist from top to bottom very, very, very tightly, and it would end up making this spiral or helix type shape. It's a double helix, meaning that it's going to go two ways. All right. And this shape of DNA was, quote unquote, discovered or given credit to two scientists of the name of James Watson and Francis Crick. They were the ones that were, again, given credit because they publicized it, but they actually stole the work from this woman named Roslyn Franklin. All right. And there's that name, Chargaff. So what Roslyn Franklin did is that she gave her life to science in a very unpurposeful way. So what she was doing over on the right of this image, that little X shape that's kind of blurry, that was the first image that we captured of DNA's structure. And that was produced by Roslyn Franklin setting up this experimental model, which had her cast out radiation, and it would hit some DNA, and then the image that was kind of like the shadow of whatever it backsplashed on is what you're seeing in the bottom right of this slide. And Franklin actually worked in the office of Watson and Crick, but unfortunately, women were not respected in the science field at that point in history. And all of Roslyn's work was essentially copied, and they got credit for all of the work. It kind of stinks, but and again, Chargaff's rule was that A's will always, or adenines will always pair with thymines, and cytosines will always pair with guanines, and this is called the base pairing rule, right? All right. So base pairing rules, again. There's a reason why I'm keeping on driving this in, because as long as you can pair A's and T's and C's and G's, all right, you pretty much understand the roots of replication. All right, so adenines are always going to pair with thymines. Cytosines will always pair with guanines. Right. And as a added challenge, the accompanied assignment for this week is vocabulary definitions, big name descriptions, and a DNA replication practice. It's all in the same document. And in the vocab words, I have the words purines and pyrimidines, and I want you to be able to figure out which ones are what. 2 of the 4 are purines, and 2 of the 4 are pyrimidines. But purines and pyrimidines will always connect. All right, and that's just based off of molecular structure. Right, and yes, they are all connected by hydrogen bonds, right? So there are many different models that are going to be able to show you DNA and such like this, all right? So over on the left, very spelled out A's, T's, C's, and G's, dashed red dots for the hydrogen bonds, and you see that 2-rung twisted ladder shape, that is the double helix, all right? And so that's kind of just noting like the actual names of the purines and pyrimidines. In the middle, very snippet, very flattened out kind of structure. Up here, just following my cursor up here on this thymine, all right, this P in that yellow circle, that is your phosphate group. This very small pentagon, that's actually your 5-sugar. And then this is your nitrogenous base with all the letters in it. And so once again, each nucleotide is made up of 3 different things: a phosphate group, a sugar, and a nitrogen base. All right, and then this is more like the actual chemical structure of DNA. All right. This Jurassic Park clip I kept in there because it actually was pretty good. And if you would like to watch it, it's just 3 minutes on an unedited portion of the original Jurassic Park. Explaining how by extracting mosquito or a blood that was in a Jurassic time mosquito, they were able to take out DNA, have its original dinosaur-esque DNA, and that's how they started cloning the dinosaurs in Jurassic Park. This is actually a cool demo. If you ever wanted to do this on your own, I might do this as a little lab demo for you next lecture video. But if you mash up some strawberry into a test tubes work best, but I doubt you have test tubes. So the smaller container preferred. But put some salt, water, and some detergent, so that's like soap, and put that mixture into a test tube. In order to separate the DNA from the strawberry, you'll have to add 95% ethyl alcohol. And the cotton-looking DNA structures will start to separate from the mixture and float to the top. And so, in short, you're just going to see like this like gooky, white, foamy bubbles just separate to the top of the test tube, and all the other non-DNA material just sinks to the bottom. Just a very simple way to extract some DNA. All right. So again, just the key points to this lecture. DNA is made up of 3 major parts: sugar, phosphate, nitrogen base. If you know that, you're good on the structure, right? We have 4 different nitrogenous bases. Again, adenine and thymine, cytosine and guanine. A's and T's, C's and G's. Know those pairs, right? Each group of 3 is a nucleotide. That just means that if it has a sugar, phosphate, and a nitrogen, it makes up a nucleotide, and the sugar is called deoxyribose. Again, OSE endings mean sugar. All right. So this is just getting a little bit into replication. So DNA replication, it makes a copy of itself. So every time we go through cellular division, we're splitting up the genetic material from one nucleus, we're copying it, and we're going to make 2 nuclei out of it, all right? And so when we had cellular division, we had 2 daughter cells. Well, before we get into the daughter cells, we got to make daughter DNA strands. And so up here in this figure, we have the parent strand or the original strand, all right? And we're going to learn that there is a enzyme called helicase that's going to break the hydrogen bonds between all of these base pairs and split that DNA structure up into 2 strands. And then we have another enzyme called DNA polymerase that is going to just start to match the complementary base pair to the parental strand that it's reading. And so say a DNA polymerase starts over here, it's going to go TAA, so it has to find complements for it, ATT. All right, same thing on the other side. All right. Yep, before meiosis and mitosis, the original strand of DNA is AATTGGCC. And so we already know, without just me progressing through the PowerPoint, that the complementary strand following this, all right, and this is part of the DNA practice on the graded work for this week. If it starts out AATT, you know the complementary strand is going to be TTAA and then go CCGG, right, like so, all right. So just a little bit on the S phase real quick. Here's that helicase. It's going to snip that double helix right in the middle, break those hydrogen bonds, and it will make what we know as the replication fork. Right? We know what a fork in the road looks like, and you can imagine how that DNA splitting up would be almost like a fork in the road. DNA polymerase, again, it's going to find those nucleotides and make that complementary strand. All right. Polymerase, there's a lot of different polymerases, and they have their own numbers of polymerase 1, 2, and 3. But essentially, there's a different polymerase that's going to go back and just read your DNA, newly made DNA complementary strand, and make sure that it was recorded and replicated correctly. Because again, we don't want mutations, we don't want mess ups and such, because it only takes us to mess up one base pair in a coding region of DNA for something to go wrong for your phenotype, right? And there's a joke up here. What did the helicase say to his buddy before he went to the bathroom? Don't forget to unzip your genes. Right, science joke stuff, I know. All right, so the rate of replication, all right? One strand is not replicated from start to finish. It would take way, 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 way too long, all right? And so if you imagine just a whole strand of DNA, it will have different parts of it, just kind of copying it all at the same time, and then all of the complementary strands kind of lift and kind of make their own combination into a chromatid. All right, Human Genome Project. This is a 16-minute video just kind of explaining on how we began to really map out the human genome and figure out how many base pairs we have in our DNA and such. And that is actually a cool hyperlink on the bottom. I'll let you discover that on your own. All right. And that's it for the slideshow. All right. So I'm going to end this video just because it's the lecture itself. And then I'm going to be posting this alongside with the PowerPoint that I was just going through, so you can have your own copy of it. All right. Can't wait to hear all your questions in the Google Hangouts. Once again, all of those hyperlinks are going to be at the top of each of your Google Classroom pages. Click on the hyperlink if you're on your MacBooks or a different laptop. And then if you're on a mobile, just click on the camera that should appear on somewhere on our homepage, and click on that, and then make sure that you have your correct settings as far as microphone and video recordings, and just come into the Google Hangout with all your questions. All right. Keep with it, guys. All right. Thank you so much for watching. And yeah, DNA is cool.